Today we're going to discuss the structure and function of macromolecules. Previously, we have been discussing atoms, molecules, chemical bonds, how chemical bonds form, and how chemical bonds break through chemical reactions. This topic in Chapter 5, Macromolecules, begins to discuss large or macromolecules that are relevant in all cells, in all living systems. We are going to see that the technologies that exist have allowed a lot of discoveries. What we're looking at here are scientists doing modeling of proteins. Proteins will be one of the four key macromolecules for our discussion. Macro, of course, means large or giant. Literally, there are thousands of atoms in a macromolecule that are covalently linked together. So stop and recall, covalently linked, shared pairs of electrons, whether equally shared or unevenly shared. The size or the molecular weight of macromolecules is thousands of daltons or kilodaltons. So we write this as K-dal or K-daltons. And we're going to try to recall that one neutron and one proton each weigh 1 dalton. Not that you would memorize the kilodalton size of the protein, but you might have a protein that's 55 kD. Or you might have a large DNA that's 240 kD. If you stop and think, what did you eat for breakfast, lunch, dinner yesterday? Often I have a student tell me they ate a bagel with cream cheese, eggs and toast and butter. What we do is we break these down into the food groups that you normally think of. Butter would be all fats. The egg would be proteins and fats. The bagel would be carbohydrates. If you put butter on it, that's fat, or cream cheese would be both fat and protein. You're probably aware that for a healthy diet, you want to balance appropriate amounts of proteins, fats, and carbohydrates. So those are the three main groups we're going to include. We will have other names for them, additional names. I start here because I think most people have heard of proteins, fats, which are also called lipids, and carbs. So we do discuss carbohydrates. We will also have the name polysaccharides. We do discuss lipids, which one type of lipids will be fats, and we'll discuss proteins, as well as the fourth one, which is nucleic acid. Quick, name a nucleic acid. Deoxyribonucleic acid, DNA, or ribonucleic acid, RNA. Those are the ones we typically talk about. Are they in your food? If you're eating a plant or an animal directly, if you're eating the muscle from a fish, or if you're eating the leaf from a spinach and the whole cell is there, yes. If you have ground up flour, maybe not. 3 of these 4 groups of macromolecules can be called polymers, which are long chains of repeating subunits. The subunits, or small pieces, are monomers. Just like we have 26 letters in the alphabet, and we can make many words, short words, long words, words such as the with 3 letters, words such as carbohydrates with 13 letters, longer and shorter words. The idea with monomers is that they're small pieces that repeat. Even if they're not exactly identical, an A is different than an E, is different than an O, or a U, or an I, they're similar enough that we can call them monomers or subunits. So polymers have many of these subunits repeated over and over. Why are there only 3 here? Which one did we lose? I'm going to go backwards. Carbohydrates, lipids, proteins, and nucleic acids are all the macromolecules we'll focus on. Carbohydrates, proteins, and nucleic acids are all polymers made up of small repeating subunits. Yes, we lost the lipids. We'll see why. First, we're going to discuss the chemistry of how the polymers are made or synthesized, or how they're broken down or degraded. If you dehydrate, you remove water. Hydration refers to water. D means remove. When we have a reaction, a chemical reaction that removes water, it's a dehydration, and that's what's going to happen when two small subunits or two monomers are joined together. So we'll take out a water. So logically, how would you do the opposite? Take this larger product, this larger polymer, and break it apart? That's right, you'd add water to break apart the large polymer into its smaller subunits or monomers. We're going to look at this on the next few slides. First with some cartoons, then with some actual molecules. So the title says synthesis and breakdown. What do you think is happening here? Which one? Monomers form larger molecules. That's synthesis by a reaction called dehydration. Some books you'll read condensation also. I'm going to stick with dehydration. But think about it. If you buy condensed soup or frozen condensed juice, how have they made that juice that you then go in your kitchen and add water to make the full pitcher of juice, or you add water to make the soup? That's right, they took water out to make the condensed product. So on the left, we have a short polymer, which is mind-boggling to me. Short defies polymer. Polymer means many. So you really need about 50 of those little purple circles. But we'll go with it. Or at least 12 or 15 to have a polymer. But the functional group on the left next to the 1 is an OH, a hydroxyl. And on the right-hand side of number 3, these are 3 different monomers. We have a hydrogen. The new monomer, which we can put a number 4 on here, also has a hydroxyl on the left and a hydrogen on the right. These are representing covalent bonds. And really what's going to be in this purple circle is going to be some carbon compound. We're just giving it a generic cartoon right now, because the part we're interested in is taking the hydroxyl from one monomer and the hydrogen from the chain of the polymer. Hydrogen plus hydroxyl, remove it, take out water. Water is one of the products of this reaction. The other product is this longer, let's pretend there's 15, polymer that is longer by 1 subunit. So dehydration removes water and makes a larger organic product. So how do you turn it around? The second half, breakdown. Let's see if I do have a pointer. Ooh, I do. Breakdown, disassemble, degrade, break apart by removing water. By adding water. Hydrolysis. You have the long polymer with, in this case, still 4 subunits. You break this carbon-carbon bond, or whatever these 2 covalently linked atoms are in the molecule. You take water, you split one of the hydrogen-oxygen bonds. One piece, in this case it's the longer product, receives the hydrogen. The simple monomer receives the hydroxyl. Hydro is water. Lysis always means break. So next we're going to look at this with a few specific molecules. See if you can answer these questions for yourself first. Which type of chemical reaction forms a large or larger organic polymer? That's right, dehydration. Dehydration synthesis. Synthesis means to make. What is the reverse reaction called? I'm sure you wrote or said hydrolysis. Compared to the forward reaction, what size are the organic products which are made from the reverse reaction? If you're hydrolyzing, you're taking a large molecule and releasing smaller molecules. So what we're going to do now is introduce each of the 4 classes of macromolecules. We begin with carbohydrates. Probably can name 4 foods off the top of your head that are carbs. Maybe you said that bagel. Perhaps you said toast. Maybe sugar that goes in your coffee or honey. All of these are sweet at some point. We are going to learn classes or subsets of carbohydrates. If you have many, 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 many small pieces chained together, they're called polysaccharides. One of those pieces would be a monosaccharide or a simple sugar. Monosaccharides or simple sugars can be 3 carbons. It would be called trioses. 4, 5 carbons would be pentoses, like pentagon, 5 sides. Hexoses, hex is 6 carbons. What do you think the 4 is? Tetros, 7 is heptos. The formula for every carbohydrate is carbon 1, hydrogen 2, oxygen 1. Notice we don't write the 1s, but carbon and water. Carbon hydrate, carbohydrate. Yes, carbon and water, carbohydrates. And it's always in the same ratio. So if you have 6 carbons, you need twice as many hydrogens and the same number of oxygens, carbohydrates. And previously, someone had called this glucose. It is a simplification to call this glucose. I found the pen dangerous, because technically, this could be any hexose. We'll see that in a minute. Aha! Monosaccharides. Don't panic, we're going to walk through it. First, look at the number of carbons. Trioses, we said, have 3 carbons. 1, 2, 3. 1, 2, 3. So what's the difference between the one on the top and the one in the middle? Let's count. There's 1, 2 hydroxyls. 1, 2 hydroxyls. Are they both in the same place? No, that's a difference. What's this other group called? On the end, it was an aldehyde, so this is an aldehyde. In the middle, it's called a ketone, so this is dihydroxy. 1, 2 hydroxys, acetone. Acetone has 3 carbons. So the position of the double bond O is significant. The position of the carbonyl group, which is either an aldehyde or a ketone, is significant. We're not going to memorize these structures. You would know that they're both trioses if you looked at them. Pentose, again, ribose versus ribulose. 1, 2, 3, 4 hydroxyls. 1, 2, 3, 4 hydroxyls. The position of the double bond O, the carboxyl group, the carbonyl group, is what's significant. Which one's in DNA or RNA? Ribonucleic acid. It's this one. That's right. This one's in RNA. A minute ago, we mentioned the C6H12O6 was glucose. Here it is. Or if you position some of the hydroxyls differently. Remember, these are flat pictures and they're really 3D. They spin all around. These 3 hydroxyls are on the right. Those 2 are on the right. and this one's on the left. Only this one has changed position. That's the difference between something called glucose and galactose. We would call these isomers. That's right, isomers. These are isomers too, actually. Glucose, galactose, fructose all have the same chemical formula. Instead of having the carbonyl on the end for an aldehyde, that's this end, on the last carbon, in fructose, the carbonyl is in the middle. These are linear molecules. Something really interesting happens when you get 5 or 6 carbons in a row. Instead of saying linear, this hydroxyl carbon can join and form a ring. And that's what's coming up next. Oh, it's the same picture. I don't know, is that clearer? Some everyday monosaccharides that you should recognize the names of, not the structures, are glucose. We will talk a lot about it in coming up chapter. It's the fuel for cellular work. Ribose, which we just saw on the last slide, is the main sugar that's part of nucleic acids, such as ribonucleic acid, RNA. We will see the importance of something called glyceraldehyde. 2 of these join to form glucose. Galactose is a sugar found in milk. These are all simple monosaccharides. And fructose sounds like fruit. It's a monosaccharide found in fruit. Here's reminding you what I said a minute ago, that this linear molecule can and does form this cyclic form. Again, we're not going to memorize these structures, but you should be aware that they exist. What do the arrows forward and backward mean? Yes, equilibrium. So the forming of the ring happens as fast as the rate of the forming of the linear. And here we have carbon 1, 2, 3, 4, 5, 6. Carbon 1, 2, 3, 4, 5, 6. 6 is coming up. 6 is a flag. It doesn't go in the ring. The oxygen's part of the ring. Often you'll see this structure, but in a minute or two on a slide, rather than me try to draw it here, you'll see a simplification of this where the hydrogens are omitted. So we just have the OH, the OH, the OH, the OH, and the CH2OH. Or if we really want to simplify and we're just doing generic cartoons, we will suggest it's a sugar. I knew I couldn't draw with this. We'll suggest it's a sugar by giving a hexagon. You'll see lots of hexagons chained together in just a second. That's why I tried to do that. First, before we make a polysaccharide or a polymer, we're going to make a disaccharide. Common disaccharides that you may have encountered, certainly you've had a glass of milk once in your life. The milk sugar is lactose. Notice it has 2 different monosaccharides, glucose plus galactose. Maltose is what forms when you join a covalent bond between 2 6-carbon glucoses. And table sugar that you put in your coffee or tea or on your cereal is 1 glucose and 1 fructose. Each one of these, you may or may not recall, are 6-carbon sugars. So how many carbons are going to be in these disaccharides? 12. Reminding you, how do these join together? Remove water, dehydration reaction. To synthesize or make maltose, dehydration reaction to make sucrose. So let's stop and review that. There's that complicated picture with 1. Oh, I forgot to say, this is a carbon. That's carbon 2. We imply they're there. That's carbon 3. That's carbon 4. That's carbon 5. The only one we drew the C4 is carbon 6, the one not in the ring. So this is glucose. We have a hydroxyl. This is a second glucose. It's matching entirely. From carbon 1, we remove the hydroxyl. From carbon 4, we remove just the hydrogen. and that dehydrates, takes out water. The reaction goes forward. So the reactants are 1, 2 glucose molecules. The product is 1 maltose. The bond is called a glycosidic link. There's a couple of bonds you have to know the names of. This is one. You can just call it a glycosidic bond and we're fine. Look, if you join 1 glucose with 1 fructose, it's still a glycosidic link or a glycosidic bond. I'm not going to worry about the numbering system here. That's if you're going on to chemistry. So how is this fructose different? Carbon 1 is here. It's not in the ring. That's carbon 2, that's carbon 3, that's carbon 4, carbon 5. Carbon 6 is not in the ring. So that little tiny difference in the ring structure gives you sucrose versus maltose. Again, you don't have to draw the structures or recognize the structures in this class. You have to know these ideas, and you need to know that you join glucose plus galactose with what type of a reaction? Dehydration. And the bond that forms is a glycosidic bond. You make a disaccharide maltose by joining 1 monomer, 2 monomers of glucose, or 1 monomer, a second monomer of fructose. So what are some common large carbohydrates? Complex carbohydrates in everyday speak. We don't just eat sugar or molasses or honey or fruit. We eat bread and grains and corn, rice, corn. Those are grains. We eat starch, plant carbohydrates. So that gives our body energy. We're going to look later in detail how that happens. You'll look at how starch is broken down in your digestive tract. When we eat the starch and our body breaks it down into glucose, which is a major fuel source, but we have too much glucose, our body takes the extra glucose and stores it, not as starch, stores it as a molecule called glycogen. You're going to see cartoons in a second. They're going to look exactly the same. I'm not going to stress the details of the chemistry of why they're different, but you should know in our bodies, particularly our muscles and our liver, we store glycogen. Name a starchy plant. Sure, potatoes, rice, anything we call carbs usually. Just like these are storing sugars, there's some sugars that are used to build things, so we call them structural. Just like we have wood to build our furniture or cement blocks to build our buildings or bricks, there's molecules that are carbohydrates that build things. Cellulose is a common one. It's what makes your apple or your carrot crunchy. And every plant has cellulose. We will see next chapter. Plants have something called a cell wall. The cell wall of every plant is made up of this structural polysaccharide. And chitin, that's a K, not a Ch, chitin, is in the exoskeleton. Does somebody know what an exoskeleton of insects? This is for in class. Sorry, I can't write insects. You can write insect in there. Okay. So I warned you, or I cautioned you, that pretty soon we would take that complicated structure. If you look carefully, these are supposed to be hexagons. And starch is the plant polysaccharide for storing energy. So there will be granules or grains of starch stored in the plant. And notice this is in the chloroplast. And in animal, let's call this, I believe it's a muscle, actually. There's chains of the similar, although not same, hexagonal glycogen. It's an animal polysaccharide for storing energy, for storing sugars. Polysaccharides for storage. Here's why it's different. I'm not going to stress that here, but if you're going on in chemistry, this linear molecule can go to the left, it can go to the right, and it's called alpha or beta glucose. Which ones you have and how they link together gives you starch or cellulose. That's all I'm going to do with you guys now. Don't panic about that. They're isomers. That's what it is. They're isomers. This shows you the same thing, and if you look at it carefully, the hydroxyls are here on the bottom, and you are alternating where the hydroxyl are. I'm not going to panic about that. The structural carbohydrate, now we're looking at a plant cell. This brown layer is the cell wall, and here's an electron micrograph of those fibers or fibrils that, if you tease that apart, are made up of chains of glucose that cross-link. It's a different pattern that makes it structural. Structural carbohydrate, cellulose, plant cell walls. That's the take-home. We don't digest cellulose. We eat foods high in cellulose and call it fiber. You eat carrots or celery. You eat broccoli or spinach. All of these things have fiber. However, cows and some other ruminants have bacteria that live in their stomach, in their digestive tract, that the bacteria is able to digest the cellulose, break it down to get energy. And so when the cow eats the grass, it doesn't use that cellulose as roughage. Instead, it gets broken down for energy. Here's an example of chitin. This is right now. They're in the news. They're coming back. What's in the center? The cicadas. I think we have the 13-year brood this time. And this is its exoskeleton. It's coming out. It's climbing out. This, don't panic about it, is the ring structure. That should look familiar. Carbon 1, 2, 3, 4, 5, 6. What's different? It has this little side group in blue. All we're going to know about chitin is it's a structural polysaccharide. It makes the exoskeletons of insects, or really all arthropods, such as a crab, lobster, insects. It's the monomer, the piece that's chained together to make the polymer, the polysaccharide. And interestingly enough, surgeons use chitin to make a very strong fiber or thread. Cool, huh? Go ahead and let you think about these.